A good afternoon to you at the Agile Conference in Bangkok. Let me start with a big thank you to the organizers and my session chairs, Professor Field and Professor Molschein, for the opportunity to participate. I will present an update on the methodology for clinical usefulness in this session, and I apologize for not being able to attend the conference in person. Life didn't follow plan. My name is Volker Liebenberg, and I work as a physician in industry. Currently, I'm Chief Medical Officer at I-ELCAP in Sweden, a small diagnostics company. Allow me to point out a bias of the presentation towards early cancer detection rather than the other indications. Biomarkers other than imaging are typically associated with pathophysiological processes and less with localization. When we look at the evaluation of biomarkers, typically we focus on performance. And in this session, we've already heard quite a lot about this. And there's no doubt that performance is important. But did you know it's actually a poor predictor of clinical utility? A recent publication describes an evidence-based guideline to predict biomarker success. The authors call it the biomarker toolkit, and they tried to predict the success of a biomarker early on using literature criteria. So they used a systematic, comprehensive review of all available papers of the diagnostic biomarkers in breast and colorectal cancers. And what they found were 129 attributes associated with the clinical utility of a biomarker. Quite a number. And what the diagram here on the right shows are the performance-related attributes on the lower part, and then how they are linked to biomarker success, comparing the performance criteria of successful, the rings, and unsuccessful, the dots, biomarkers. And the only one highlighted here, which was statistically significant, was the negative predictive value in breast cancer. All other weren't statistically significant of a predictor for future success. And notably, the total number of successful biomarkers, so the rings that you see across here, is actually pretty low. And these two indications are the most established ones in screening and very frequent cancers. To better understand this, it makes sense to have a closer look at clinical utility and what it means. It's defined by the NCI as the likelihood of a test to inform clinical decisions that eventually improve patient outcome. In previous presentations talking about analytical validation, clinical validation, and positioning, however, clinical utility extends beyond these aspects to include regulatory approval, cost effectiveness, and then ultimately the acceptance by the medical community. So a rigorous assessment to provide the solid evidence of effectiveness of a biomarker needs to answer additional questions beyond what we've seen before. So it's about the reliability of the results, it's the impact on the clinical decision-making, it's the potential harm which drive the need for clinical studies. The methodology for biomarker evaluation is at the core of such assessment. There are multiple frameworks available for structured development and evaluation, but for today, we have not enough time for a detailed discussion of those. The most comprehensive academic framework has been developed by the Early Detection Research Network, or EDRN, over the past about 20 years. It outlines 5 key phases. Each comes with detailed protocols to evaluate a biomarker stepwise. And for a biomarker to demonstrate clinical utility, it actually must successfully complete all phases, particularly analytical and clinical validity that we heard about, and then clinical utility and patient impact. Now, this 5-phase approach appears to be comprehensive, and yes, it mentions clinical utility. Nevertheless, it's quite a difference between generating such evidence from clinical studies and then obtaining regulatory approval and reimbursement based on cost effectiveness. These are typically commercial activities, and if you want to pursue them as a company, you need to be ISO certified and perform health technology assessments. Now, the scientific rigor, together with the requirements for the authorities, have made the cost and effort for biomarker development comparable to that of drug development, which unfortunately, if you compare the commercial attractiveness, drugs are far more interesting than biomarkers. On a high level, both academia and industry share the same goal of improving patient care, but actually they operate differently. Academia prioritizes scientific discovery, novelty, and publications, and controls access to patients. Industry, on the other side, focuses on rigorous validation, compliance, and commercialization to ensure economic sustainability. Now, these two come from different directions, nevertheless. They happen at the same time. And what they lead to is collaboration out of necessity rather than by design, and a low efficiency in biomarker development based on the level of validation and limited reproducibility. Now, together, this increases the cost of development, especially in combination with the well-known biomarker challenges. And what we see as a consequence is a low rate of success, as you already saw in the low number of successful biomarkers at the beginning. Keep in mind, blockbuster biomarkers like troponin or BNP emerge only about once a decade. So the key challenges that come on top are, of course, well known. It's the variability due to biological complexity. It's the regulatory requirements, particularly if you're looking at a novel biomarker with a new scientific rationale and a new technology. Then you need to integrate this into the workflows, which are heterogeneous. And finally, the more we become in the world of precision, the more privacy concerns become relevant. So this all adds up, and then what people underestimate is the cost of the final step of real-world translation. So failures that happen there are very costly and then reduce the willingness for future investments. To illustrate how this looks in real life, if you consider a new biomarker, I want to show you a brief high-level case study for a biomarker utility. It illustrates an optimized approach in 7 steps to evaluate a biomarker for early cancer detection. The intended use was picked for a risk marker, which actually is the lowest performance requirement of all diagnostic markers. Then technology development has been started early with analytical validation to make sure that the results are reliable. The indication comes with strong evidence of outcome benefit. From risk-based testing, what we're seeing is implementation already of risk models in lung cancer. Now, the intended use from risk is formally non-actionable, which means from a regulatory perspective, this is a moderate risk device. Again, the lowest possible category that you can do there. In parallel, health economics is done and the test cost is modeled so that at any given time, it's clear how the commercial situation would potentially look like in future. And then finally, guideline inclusion is pretty likely based on comparative performance due to all the evidence available. Now, if you put this all together, it's still a 10-year process for a new biomarker. And it took about the first 5 years for the technology development, and the second half will then be on the clinical validation and the utility. And a key lesson learned is that then halfway through the process, we realized that none of the existing lung cancer studies available in the world actually meets the FDA requirements to approve or clear a product. This alone has the potential to extend the timeline by another 5 years. So in summary, it's great that academia and industry share the same goal of developing clinically useful biomarkers and improve patient benefit. The methodologies we have for rigorous evaluation are established, and it's very common already to cooperate around clinical studies. Now, rather than summarizing all this, if we look at the outcome, we realize this is not enough. So where do we see the need for change? I think we need to get better at planning for biomarker translation from start. The scientific methodology for clinical utility needs to think about and prepare for the requirements of real-world implementation. I see huge benefit if such collaboration between academia and industry would start way earlier. We need to align the processes of the stakeholders to increase the efficiency and reduce the probability of failure at the late stages. And this is really an urgent need because, as we all know, precision medicine will continue to raise the bar. Agile is already asking for 3 biomarkers for the big 3 indications, and it's unlikely that 1 biomarker can do it. It might not even be 3, which are enough. And then multi-cancer detection, targeting up to 14 cancers in parallel, is around the corner, and the complexity is even higher. So let me close with a final thought. We all rely on AI more and more, so I thought I'll ask Gemini for a creative idea how to deal with this, and it came up with an analogy saying, if you struggle to catch one ball, focus on improving your catching skills before attempting to handle multiple balls. Thank you for your attention. I look forward to the discussion.